Alright, so welcome to our today's lesson where I'll be talking about glycogenesis. Okay, glycogenesis. Just from the name itself, glycogenesis, what are we making here? We are making glycogen. Okay, so you guessed right. So we are making glycogen. We are making glycogen because the genesis means we are formation of something, and glyco means or came from the word glycogen. Okay, so we are producing glycogen, but from which molecule? From glucose. Okay, so this is basically a glucose molecule. Okay, so now where are we making this same glycogen, and what is the reason of us making this same glycogen? Okay, so where we are making the same glycogen, it is of course in the muscles and in the liver. Why are we making the same glycogen? It is a storage form of what? Glucose. So what we are trying to do here, we are trying to store what? Glucose, okay, for later or future use. So that whenever we're starving, that glycogen can now be converted back to glucose. Okay, so that now our body cells can start using what? The same glucose that was stored in form of what? In the form of glycogen. Okay, so here, this cell that we're able to see here, it is a liver cell. So this is a liver cell. So now in the liver cell, we have some channels or glucose transporters that are responsible of transporting glucose inside the cell, okay, inside the liver cell. We call this glucose transporter what? 2. If it was in the muscles, then it would have been what? Glucose transporter 4. Okay. So now here, let us try to make glycogen we see. Okay, because this is a very funny process. Why? Because here you are going to find a lot of enzymes arguing. Okay, this does this, this does this. Okay, enzymes will be doing what? Will be arguing more like they are playing. Yeah. Okay, so now this is a glucose molecule. So that glucose molecule will enter this liver cell so that now it gets to be converted to which one? To glycogen. Okay, so now if it enters, okay, let's see it has entered now. 1, 2, 3, 4, 5, 6 carbons. Okay, this is carbon number 6, this is carbon number 1. So that glucose has now entered through which transport channel? Transport channel, which is glucose transporter what? 2. Okay, it has entered inside the cell. So now what will happen to this glucose? Because it has got a tendency of going back again. Okay, so immediately it reaches the cell, there's actually an enzyme that is responsible for not allowing this glucose molecule to go back. Okay, so it will prevent glucose molecule to leave the cell again, so that it gets to be converted to what? To glycogen. So now that enzyme, because we're talking about the liver cells, that enzyme we call it glucokinase. Glucokinase. So glucokinase. Okay. In our previous video, I said that kinase is actually an enzyme that carries what? A phosphate group. So even this one, it is carrying what? A phosphate group. Where is it bringing, or where is it taking that same phosphate group? To the glucose. Why? So that it binds the glucose, so that glucose won't go away or won't go out. Not until it gets to what? To be converted to glycogen. You see? Okay. So this enzyme is very much important. As you can see, it is actually allowing this same glucose to stay here so that it gets to be converted to what? To glycogen. Okay, so that enzyme is glucokinase. So now it is bringing the phosphate and attaching that phosphate to what? To carbon number 6 here. So it is bringing the phosphate group and putting it at carbon number 6. We're going to call this molecule glucose 6-phosphate. Why 6-phosphate? Because at carbon number 6, we are having what? A carbon, or rather, we are having a phosphate group, okay, that is attaching there by the help of what? Glucokinase. Okay, so this is now the glucose 6-phosphate. Okay, glucose 6-phosphate. So that is actually the molecule that we have yielded here. Okay, just by the addition of this phosphate, even the name of the molecule has changed from glucose to glucose 6-phosphate. Very easy. Okay, so now this molecule that we have here, the glucose 6-phosphate, what it will do now, it will be changed again. Okay, it will be changed. Now here, the play now has started. Okay, by the what? By the enzymes. You will see this enzyme, the glucokinase, was smart enough to bring the phosphate group and put it to carbon number 6. But again, there's another enzyme that will come and argue to say, no, 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 it doesn't work like that. We can't put it carbon number 6. Let us put it carbon number what? Carbon number 1. Okay? So the phosphate will be shifted from carbon number 6 to carbon number 1. Okay? So 1, 2, 3, 4, 5, 6. So at carbon number 1 here, we're going to have what? A phosphate now. That phosphate that we had here has now shifted to carbon number 1. Okay? By which enzyme? So that enzyme that argued to the glucokinase, we call it what? We call that enzyme phosphoglucokinase. Okay? Rather, we call it phosphogluconutase. Okay? Because this is mutating, it is not carrying a phosphate. So we can't call it a kinase, but rather phosphogluconutase. It is now mutating. What is it mutating? This molecule, the glucose 6 phosphate. Okay? It has been mutated. As you see, this group was at carbon number 6, but it has shifted to carbon number 1. Even the molecule has now changed from glucose 6 phosphate to what? To glucose 1 phosphate. Why? Because at carbon number 1, that is where the phosphate is now attaching. So this is glucose 1 phosphate. Okay? Glucose 1 phosphate. Okay? So this is a very important intermediate. This is very important what? Intermediate in our process of making glycogen. Okay, so we are still going. This is a very important intermediate. So what it will do, the glucose 1 phosphate that has been made by that enzyme, which we are calling phosphogluconutase, this molecule, it will now be combined to another molecule that is there present in the cytosol of liver cells. We call that a what? Uridine triphosphate. So this is a molecule that will now be combined to what? To glucose 1 phosphate. Okay? Uridine triphosphate. So this is a uracil plus a ribosome. We call that a what? Okay? A uracil plus a ribosome. If we combine uracil and ribosome, we call this uridine. Okay? So this is basically uridine. No wonder this molecule that we are seeing here, it is uridine triphosphate. So in short, this uridine that we are having here, it is actually connected to 3 phosphate groups. So we are having that 1, we are also having another 1, 2, then 3 like that, inorganic phosphate, and attaching to what? To this molecule, which is a uridine. Okay, so this we are having here, this molecule, this is uridine triphosphate with how many number of phosphate groups? 3. 1, 2, 3. Okay, so now what will happen to this molecule that we have here? It will now be connected to what? To glucose 1 phosphate. Okay, so now immediately it connects, it gets to be attached or reacted with glucose 1 phosphate. This bond here that we have here, it will be broken. Okay, that bond. So meaning these 2 phosphates that we have here, these 2, they are going to be separated from the uridine triphosphate, and this molecule now we're going to call it what? Uridine monophosphate. Why monophosphate? Because only 1 phosphate is remaining here and attaching to what? To the same uridine. Okay, so what we're having here, it is uridine monophosphate. Okay, this is what we're having here. Uridine monophosphate. We're just having 1 phosphate group here, which is this one, and these two phosphate groups, even their names now, they have changed. We call them pyrophosphates. Okay, these 2, they are what? Pyrophosphates. So let me just write it here. These 2, they are pyro. Okay, pyro, then phosphate. Okay, but why are we calling them pyrophosphates? Okay, just from the name, as you can see, I've written the word pyro in red to symbolize that this pyro means fire. Okay, the word pyro, it means what? It means fire, the same fire that we know. Okay, it means fire. But why are we having a fire phosphate here? So it is just basically symbolizing to say that these these phosphate groups that we are having here, they are actually high energy. There's actually a high energy bond in between there. Okay, there's a high energy bond, and if that bond is broken, it will release what, a lot of energy. Okay, and we know that this process requires what, energy. Okay, it requires energy. It is endergonic process. Okay, so it is absorbing what, energy for it to what, to complete its process. Okay, so now these two, they are pyrophosphates. Why pyrophosphates? Because they are high energy carriers. Their bond, it is carrying a lot of what, energy. We call that bond as phosphor phosphor hydride. Okay, hydride, like that, anhydride. So this is a bond that is pyro, or that is fire. It has got a lot of energy. Okay, if we break this bond, a lot of energy will be what, will be released here. Okay, so now what will happen is, because this process requires energy, so it's very important for us to break this bond. Okay, so that we also now facilitate the combination of glucose 1-phosphate with the uridine monophosphate. Okay, because we are remaining with what, just 1-phosphate. So here what we're having is uridine monophosphate. Okay, so what we are having now is this glucose 1-phosphate that you are seeing here, it will now react with uridine monophosphate. Now, uridine monophosphate, it is having a what? A monophosphate, 1-phosphate group. So that 1-phosphate group, it will now react with this phosphate that we are having here. So that uridine, it will now become what? Uridine diphosphate. Uridine diphosphate plus what? This glucose molecule. Okay, so it is uridine diphosphate glucose. Okay, we can even term it like this. This is uridine diphosphate and glucose. Okay, so that is now the molecule that we have there, uridine diphosphate glucose. But from here, we can't continue minus having what? Energy. So this is where now energy is required. Okay, for this process to continue, we need what? Energy. Where is that energy going to come from? From, of course, that bond now, the pyro bond, that bond that we talked about, from pyrophosphates. Okay, so when we break this bond of Pi, which is monophosphate, again Pi, which is another monophosphate. So if we break this bond that we are having here, energy will be what? Released to the environment. We are breaking that bond. What enzyme is breaking that bond? Okay, the enzyme that we are breaking this bond is very simple to know, because these two, they are called what? Pyrophosphates. And breaking these two bonds, or this bond, by an enzyme, it is what? Pyrophosphatase. Okay, this is pyrophosphatase. Okay, it is this enzyme that is facilitating or stimulating the breaking of this bond in between here. So that now, if we break the bond like this, we are now going to do what? To yield energy. Okay, so the energy now will come there and help now do what? Start now the process. Okay, continue with the process of UDP plus glucose, so that we form what? Glycogen that we need. Okay, so that we store the glucose in the form of what? Glycogen. This is the whole part of this process. Okay, so now phosphatase, pyrophosphatase has now done the needful and has broken the bond here, and the energy has been yielded. That energy will be required to drive this process. UDP plus glucose will now continue with its process. Okay, so what we are having here now is UDP plus the glucose. So what will happen with the UDP plus the glucose that we have here? So the UDP plus the glucose, they are now going to react with another molecule that is present right there in the what? Liver cells cytosol. Okay, so that molecule, we call it what? Glycogenesis. Okay, this is a molecule that is present there in the what? In the cytosol of liver cells. Okay, glucogenesis. Now, the funny part of this glucogenesis, it has got, Okay, it has got a molecule on top of it, more like a protein. That protein, we call it what? Tyrosine. Okay, so this is a glucogenine. That's glucogenine. It is having what? A tyrosine. So it is having a tyrosine molecule just on top. Okay, like that. Let's say that is a tyrosine connected to glucogenine. So this glucogenine now, it is actually our what? Our foundational thing here that will be used in order to make the same glycogen that we need. Okay, it is more like a foundation. Okay, so what we're going to be doing is we're going to be getting a lot of UDP plus the glucose, because imagine this process is happening like simultaneous. A lot of glucose now, they are being brought so that they get to be to be converted to which one? To glycogen. Okay, this is what we are doing here. A lot of glucose will be brought inside the cell. A lot of glucose will be brought inside the cell so that we get now to convert those glucose to what? Glycogen. We store glucose in the form of what? Glycogen. So now here, this glucogenine that we have here, since it has got this tyrosine, it is now where the UDP and the glucose will be attached. Okay, so the moment the UDP and the glucose is attached here, so what you need to note here is that this UDP is just allowing for the what? For the movement of the glucose here. Because the moment we put a glucose molecule here, the UDP will be released to the environment. Okay, in short, if we put a glucose molecule here like that, glucose molecule, it won't be glucose plus UDP. Why? Because this UDP would have been what? Released to the environment. This is what is happening. So the moment we add the glucose, the UDP is now released to the environment. UDP. Okay, like that. So now here, there are some terms that you are supposed to know. Okay? There are some things that you are supposed to know. So look, what we are doing is this. If we have a glucose molecule, like it is having a glucose molecule, like maybe a structure itself, it is like this. So if we draw a glucose structure molecule, okay, like that, then okay, so this is 1, 2, 3, 1, 2, 3, 4, 5. Okay, so they're supposed to be how many? 6. Okay, so this is now what we have here. Okay, like that. So we have carbon number 1. Where are we having carbon number 1? Carbon number 1 is right here. Carbon number 2 right here. Carbon number 3. Carbon number 4. And carbon number 5 here. So carbon number 5 here. But at carbon number 5, we are also having what? Carbon number 6. So at carbon number 6, it is found where? Just on top of carbon number 5 here at this group, CH2OH. Okay, so this is carbon number 6. So now, basically what is happening here is, when we have this glucogenine, so glucogenine, this is now where this guy, the glucose, will be now attaching to another glucose. So this glucose, where we have carbon number 1 here, let's say we have carbon number 1 there, okay? So carbon number 1, if we try to add another glucose molecule, because this is what we're going to be doing here, we're going to be adding glucose on glucose, glucose on glucose, so that we accumulate a lot of what? Glucose, so that we store them now. So now here, at carbon number 1, since we have the carbon, that will now be attached to another glucose molecule. So let us also try to do what? To draw another molecule of glucose like this. Okay? Like that. So if you see closely, this is carbon number 4. And this should be what? Carbon number 4. So this is carbon number 1, 2, 3, then 4. So whenever you have a carbon number 1 and carbon number 4 attaching like this, and of course, if maybe the hydroxyl group, they are also pointing upward in the same direction, we call this what? Alpha bond. Why? Because the hydroxyl group, they are what? Being oriented in the same side. Okay? They are pointing upwards, both of them. Now we are having a bond here, which is happening between which one and which one? Between carbon number 1 and carbon number 4. And this is what? Alpha bond. Why are we calling it alpha? Because the hydroxyl, they are all pointing where? Upward. So this is alpha 1, 4. Why 1? Because it is between carbon number 1. Why 4? Because at carbon number 4 it is also now participating in this bond. So it is carbon number 1 and 4 which are fusing here. So it is alpha 1, 4 glycosidic bond. So this is glycosidic bond. Glycosidic bond. Okay, that is the bond that we have there. So now what we're going to do is, from this knowledge that we have here, we're going to be now adding glucose on glucose, glucose on glucose. So whenever we add a glucose on another glucose, if we add the glucose to carbon number 1 and carbon number 4, then we're going to form which bond? Alpha 1, 4 glycosidic bond. What if we add a glucose molecule at carbon number 6? Because even at carbon number 6, we are also going to have what? Another bond. So now the moment we have, so carbon number 6, it is where? Just here on top. So this is carbon number 6. So let's say at carbon number 6, we try to add another glucose molecule, okay? What will happen is we're going to say, this is carbon number 6. So we are going to write like that. So here we are supposed to have that oxygen, then that, okay? If we say that this is, okay, so the oxygen should be this side. So if we have like this should be what? Carbon number 6. So at carbon number 6, since we are also having a bond, but same bond and same orientation of these hydroxyl groups, this bond, you're not going to call it 1, 4 glycosidic bond. Why? Because it is not at carbon number 4, but at carbon number what? Carbon number 6. It will still remain alpha, but 1, 6, same glycosidic bond. Okay? Glycosidic bond. Like that. So now, this from here now, we are going to build what? Everything. So we have seen that the UTP here and the glucose have been combined together, and the glucose now is brought to where? To this molecule, which is a glycogen. Okay? It is this protein which is having a tyrosine. And it is there on the tyrosine, we're now going to do it, to be adding the same glucose molecules. Okay? So we're going to be adding a lot of glucose molecules. They are supposed to be how many? 6 glucose molecules. 6 glucose molecules at first, because these are going to react as what? Primers. Okay? As primers, because that is now we are going to be doing what? Adding a lot of glucose just on top, adding until we yield what we need, which is what? Glycogen. Okay? So we have the UTP and the glucose there. So the glucose has now been what? Been added on the glycogen. So from the information, from the knowledge that we have there, so we said at carbon number 1 and 4, this we are forming what? Alpha 1, 4 glycosidic bond. What if at 1 carbon? If it is 1 carbon and at carbon number 6, so this is 1, 6, we are forming the same alpha, but 1, 4 glycosidic bond. So between carbon 1 and 4, we are forming alpha 1, 4. Then between 1 and 6, we are forming what? Alpha 1, 6 here. Okay, 1, 6 glycosidic bond. Yeah. That is basically what we're forming there. So now let us see what will happen there. What will happen? Because we have to form actually a lot of glycogen. We have to store a lot of glucose in form of what? Glycogen. Okay, so we have this molecule here, which is waiting for us to do what? To start now adding them. So this is glycogenin. So let me even write it here. So the glycogenin is just below here. So this is a tyrosine where I'll be adding the glucose molecules. So this is a glucose molecule, another glucose molecule. Where are these glucose molecules coming from? So they are coming from the what? From the food that this person is eating. Okay, another glucose molecule, another glucose molecule. So 1, 2, 3, 4. Let's make it 6, 5, and then 6. Okay. So these glucose molecules, they are joining together by which bond? By 1, 4 glycosidic bond. Okay. So the bond which is in between this glucose molecule and this glucose molecule and this glucose molecule, it is 1, 4 glycosidic bond. Okay, glycosidic bond. Okay, it is not this one. So this is basically a molecule. This is a protein plus a glucose molecule. They are not forming a bond as 1,4 glycosidic bond. Okay, so now this tyrosine is connected to which one? To glycogen. So down here there is what? Glycogen. So this is a glycogen molecule. Yeah. So now, since we have added a lot of glucose molecules here, we have formed 1,4 glycosidic bond. So this was happening by the help of which enzyme? So no enzyme was helping this, but by the help of the energy that was made by the breaking of what? That pyrophosphate. Okay, pyrophosphate bond that we had. Okay, so now what will happen here? Since we've made this, since we've created this, we've added glucose molecules, glucose molecules, glucose molecules, glucose molecules. So now inside this, besides, like in the sides here, we are having a carbon number 6. Even here, carbon number 6, carbon number 6, carbon number 6. So you can even tell to say here at first we are having what? Carbon number 1, then here it is carbon number 4. Okay, so the carbon number 6, it is in the sides. So now what will happen is, since we've made this, so this is more like a what? A starter or a primer. That is now where another enzyme now will come and add a lot of what? Glucose molecules. So in short, that energy that was yielded from the breaking of that bond has helped the cell by itself to start adding these glucose molecules. And of course, have you seen, it has added up to 6 glucose molecules. So now another enzyme now will come, which is responsible of adding these glucose molecules. Okay, so that enzyme will just help it, and that enzyme, we call it what? We call it glycogen synthase. Okay, so glycogen synthase, that is basically an enzyme that is responsible of adding glucose molecules there on top, so that we form what? We form glycogen. So now look, if this enzyme comes, okay, so now if this enzyme comes, which is glycogen synthase, so that is a glycogen synthase there. Okay, it will come and add what? It will come and add the UDP and the glucose. Okay, it will be carrying the UDP and the glucose, because at first we had the UDP and the glucose, and the cell itself had that power to do what? To start adding the same glucose molecules. Why? Because of the same energy that we got. So now this same enzyme, the glycogen synthase, because it is having the UDP and the glucose, it will now start adding them here. In the process, what are we going to be yielding? We're going to be yielding what? UDP. So here, the cell was adding glucose, we were yielding what? UDP, UDP, even here, UDP, okay? UDP, UDP like that. So everywhere we were yielding what? UDP. The UDP was just helping the glucose to be moved to what? The tyrosine, okay? And it was now yielded. It is now present in the cytosol of what? Of the liver cell. Okay. So now this enzyme now has come, which we call the glycogen synthase. Just from the name itself, it is synthesizing the what? The glycogen. So it will now start adding a lot of glucose molecules. It will add until it reaches level, about level, somewhere there. So 1, 6 plus this, 7, 8, 9, 10, 11. So another play now starts. Okay, we saw another play here by the enzyme glucokinase, and of course that enzyme that we had, which is phosphoglucomutase. So now here, what will happen is, this enzyme, the glycogen synthase, since it is adding a lot of glucose molecules, just it is piling them, like piling them. Now, do you think a cell will accommodate all these glucose molecules just like this? No, of course, it can't do that. Okay, so there comes another enzyme. So what it will do is, it will try and splice off this long chain. Okay, it will come and splice off this long chain and add it to where? To the other bond, okay? So it will try to shift this glucose molecule which is being aligned, which is being made along. It will come and shift it and put it where? Here at carbon number 6 in the sides, okay? So it will be like that, so that it creates more space. Yeah, so this enzyme is now thinking that no, a cell can't actually accommodate a lot of this thing that you are making, a lot of long chain. So let us try to do what? To fix them. Okay, so this is what we are doing here. We are trying to power them up, okay, so that they create more space for a cell, so that a cell now can accommodate this same what? These same molecules. Okay, so now these guys, they have been what? Spliced off. Okay, so the enzyme here was the glycogen synthase. Glycogen synthase. It was responsible of doing what? Responsible of bringing the same UDP plus the glucose molecules, where on top there it was now piling them up, piling them up, piling them up. Okay, the UDP and the glucose. Where do they come from? The enzyme itself has got what? The UDP and the glucose. Okay, it was just there to do what? To add them up, add them up, add them up. Now, what that enzyme will do, that will come and split these molecules which are going there, okay, which are making that long chain to go up. It will come and change now the position at which these molecules, they are being added. Okay, instead of putting them at 1,4 glycosidic bond, they are now going to put them where? At 1,6 glycosidic what? Glycosidic bond. Okay, it is now changing from 1,4 to 1,6. Okay, so that more space will be created. So now, what do we call this enzyme that is doing all this work? Okay, that is arguing with this enzyme, which is the glycogen synthase. Okay, it is arguing the glycogen synthase. You say you can't be adding a lot of glucose molecules. Okay, you can even see that no, this room is what? It's very small. Okay, this room, meaning the cell. So a cell can't accommodate that same long chain which this glycogen synthase is doing, what is synthesizing or it is making. No wonder that enzyme now will come and do it and splice it off and put it where? There on the sides. Okay, and that enzyme will come and put it where? 1,6 glycosidic bond. Okay, so the enzyme that will come and splice off, it will make a bond which is 1,6 glycosidic bond. What do we call that enzyme? Just from itself, look, that enzyme is making what? A branch. Okay, it is creating this branch. That enzyme, we call it a branching enzyme. Okay, branching enzyme. It is basically a branching enzyme. Okay, that is the name of the enzyme. But this enzyme, it has also another name, a very fancy name. Okay, that name we call it what? Coxyl. Okay, it is coxyl. Yeah. So coxyl, open bracket, then you say 1,4, then 2, 1,6, cross bracket. So 1,4 to 1,6 glycosidic bond. So glycosidic bond, then close bracket, then you say transferase. Okay, that is the fancy name of this same branching enzyme. But even this same one is correct as well. You can just go by that branching enzyme. Okay, it is even very easy to remember it, because it is creating, or it is doing what? It is making a branch. Okay, so now from here, another game will now do what? Will continue. Okay, another play will do what? Will continue with this same enzyme. Okay, just look here. This branching enzyme came and spliced off, okay, that long chain and added it where? To the sides, and made that other chain to be there. But again, this enzyme, the glycogen synthase, again, it will now see that, oh, there is actually more space here. Let me put another layer of what? Glucose molecule, so that we power up what? Glucose, so that we store what? Glycogen. Okay, we are storing glucose in form of what? Glycogen. So this enzyme will now come and put more of the glucose molecules, which are having what? A UDP. Okay, so more of them will come now here again, because the glycogen synthase has seen that there's more space there. Okay, Again, that enzyme will also now come and splice it off, the branching enzyme, and splice it off and put it where? Here. If there's maybe a carbon 6 there, again, it will now continue like that. Okay? So the process continues just like this until we store what? Glycogen. So this thing that we are making here, it is called what? So this thing that we have made, we call it glycogen. Okay? Glycogen. So this is the whole process of what? Glycogenesis. Okay?